I have a few slides to present to you all. Thank you for joining. I don't know what number we are at, but I'm very pleased to welcome Pablo. Now I practiced pronouncing his last name, Nepomnaschy. It's okay. As you all, if you've joined us before, will be aware, please ask questions through Slido, which you can Google it, Slido. And the event code is 55183. It should be in your invitation. I would like to acknowledge that we're hosting this event on the traditional ancestral and unceded territories of the Coast Salish peoples, including the territories of the Musqueam, Squamish, and Tsleil-Waututh nations, and we thank them for the ability to work, live, and play on their beautiful lands. I just wanted to do a brief shout out for Women's Health Research Cluster. Even though we're based at UBC, we're trying to go international. We have over 200 members right now, so we need to update our infographic. We're a multidisciplinary collaborative network of women's health researchers and stakeholders. We have pretty good diverse group. One area where we need a little bit more diversity is more male researchers. And so that's one reason why I'm very pleased to have Pablo speaking today. And really our mission is to advocate for more women's health research and promote and catalyze impactful research on women's health. And the reason why, I forgot that I had these slides in here, but there are very few studies on women's health and women specifically. You can see in animal studies, it's about 6%, and in human studies, it's around 4 to 3%. And it's not getting any better. So that's 2009 to 2019, and that's across 2010 to 2014. We also are running a Women's Brain Health virtual conference. We have monthly events, so please join us. Any trainees on the call, you can get it does cost a little bit of money, but you can get it for free as long as you apply for the funding that we have available, and we thank our sponsors for that event. For today's conversation, you can join us on Twitter. Remember the Slido code 55183 to ask questions. You can tag us if you want and join us, and you can do that actually on our website. And if you like what you see, please consider donating to support us, because we are on a shoestring budget. Now, without further ado, I am going to stop sharing my screen so I can properly introduce Pablo Nepomnaschy, who is an associate professor in the Faculty of Health Sciences at Simon Fraser University. He obtained his first degree in biology in his native country, Argentina. I can't wait for you to hear his accent. From the, I'm assuming, beautiful University of Patagonia. For his doctoral studies, he moved to the USA, where he obtained a Ph.D. in Anthropology and in Ecology, or the National Resources and Environment, from the University of Michigan. He also received training from the Reproductive Sciences Program there, and he's an alumnus of the LIFE program, a life course, Evolutionary and Ontogenetic Dynamics, from the International Max Planck Research School in Berlin. And he received post-doctoral training in the Epidemiological Branch of the National Institute of Environmental Health Sciences. So you can see he is truly multidisciplinary, transdisciplinary research. So his academic interests, and you'll see it in his talk, center on the effects of stress on women's reproductive physiology, as well as growth, development, and health of their children throughout the human life course. As you probably also know, I asked for a fun fact, and Pablo's fun fact, which I think is fantastic, is that in a previous life, before his academic career took off, he sold lipstick and lip liner to support himself while he was supporting his undergraduate degree. And he had some rocky start to his sales career, but it picked up despite never having used those products in his life, which made him realize that he's been mansplaining for a long time, which is totally fine. That's great. Thank you, Pablo. We do welcome your voice, so thank you so much. And I'm over to you.
Thank you very much. Let me see if I can share my screen. Okay. Hi, everybody. It is a pleasure and an honor to join this seminar series, and I'm grateful to everybody at the cluster for the invitation. Please send questions. I'm going to present, because this is my first interaction with the people at the cluster and this seminar. I thought I would share an overview of the main projects that we are doing in my research team. And so we will touch upon several fields as we go along. And I would like everybody to understand. And if you don't, I would not mind at all if you send a question. And I have told Lisa that I don't mind if she wants to interrupt me to clarify concepts. And some people say I have an accent. I don't know why, but if you don't understand my accent, I don't mind trying to explain the same concepts with different words where my accent may not be that thick. All right, so let's start by saying that yes, initially my career started as a reproductive ecologist. I have a very strong background in biology, but as I move along, I move into anthropology and epidemiology, and that's kind of the main message that I want to send to you today, is that women's reproduction or reproductive health can be understood from very different angles, and that those angles can provide very interesting insights. So let me see why I cannot. Okay, here. So about 20 years ago, I was at the University of Michigan working with Beverly Strassman, who was my mentor at the time, and she was working with another woman called Donna Bird that became my postdoctoral mentor. I didn't know that at that time, but they were working on these super interesting graphs. And this graph shows that if you take 100 women, 100 sexually active women, and you look at 100 menstrual cycles from them, at the end of those 100 menstrual cycles, you will end up with 23 live births. And that's because a number of them will not ovulate, a number of them will not conceive, a number of them will lose pregnancies in the 40 weeks that gestation takes place. So at about the same time, the next year, I don't know why I cannot just change slides here, like with my cursor. Anyway, the following year, a very famous OBGYN, whose last name was Norwitz, published in the super influential New England Journal of Medicine an editorial that contained this sentence: human reproduction entails a fundamental paradox. Although it is critical to the survival of the species, the process is relatively inefficient. This pathology paradigm of women's reproduction really bothered me. The moment I read it, I felt that this was a very misogynistic statement, and it had nothing to do with what I had learned about female reproduction. Because what it implies is that women are broken by design. And as I said, it just did not bode well with my training as an ecologist and as an evolutionary biologist. Coming from the world of non-human reproductive ecology, in a previous life I used to study these animals, elephant seals, I knew but one thing: the reproductive traits are always under extremely strong selective pressure. So there are no space for system-wide inefficiencies. When you see a system that results in the same outcome over and over and over, you have to think that that's the optimal outcome given all the constraints, environmental and species constraints of the system that you're studying. So that's when I was starting my journey on the field of human reproductive ecology. And for those of you who don't know this field, you can Google it and you will see that it's a field that has several decades old, and it provides a very different alternative to the pathologizing approaches that sometimes we see coming from traditional Western medical or clinical approaches to women's reproduction. And to think about women's reproduction from an ecological perspective, you have to consider one key unique factor. Unlike most other mammalian females who ovulate in spring or summer, or have induced ovulation, given the presence of males or the abundance of food, etc., women present continuous ovarian cycles. It's one cycle after the other, after the next, after the next. And so within that context, given that system based on continuous ovarian cycles, reproductive losses may not be reproductive failures, may be the result of reproductive suppression mechanisms that have helped women over our evolutionary history to regulate the timing of reproductive events. And whenever I get to this slide and I have a rambunctious room, women look at me weird because they say, well, why do we use contraception? It's a long story, but we don't live today in the world that we used to live. And I'm happy to entertain that question now or at the end of the talk. At the center of those regulatory mechanisms are these two axes, the hypothalamic pituitary adrenal axis and the hypothalamic pituitary gonadal axis, which in the case of women is the hypothalamic pituitary ovarian axis. And these two axes interact with each other, regulating when women ovulate, when they conceive, when a fetus implants, and whether or not that fetus will survive. And now we know all of this. But when I started, most of the research that was out there in terms of, for example, stress causing miscarriage was coming from animal studies. And as I just said, women are not a regular mammalian female because they ovulate all the time. So I set myself, I decided to check whether or not stress could affect ovarian function in women. And this was the first paper I wrote on the topic, where I looked at variation in cortisol levels within a woman, so variation in cortisol across days, and ovarian function. And in this case, I'm going to present many articles or many studies. I'm just going to go over very quickly, but if you want details on any of them, I'm happy to answer questions either now or at the end, as I said. So in this paper, we show that when cortisol varies, when cortisol goes up within given woman's baseline, her progesterone levels during the luteal phase, for example, are lower. So higher cortisol reduces levels of this hormone progesterone that is absolutely paramount for the development of the endometrium, the inner lining of the uterus, and critical for implantation. And then we moved on to look at the levels of cortisol women experience after conception and the risk of miscarriage. And we found that women whose cortisol levels increase by 2 standard deviations also experience almost 3 times the risk of losing that pregnancy that women who stay within their own cortisol level, baseline cortisol levels. And as many of you know, research begets research, because as you study a system and you answer some questions, new questions emerge. And one of the questions that emerged for us was what's going on with the amount of variation in terms of the variability between individuals in terms of the functioning of their stress axis. Because in order to do the studies that I just showed you, we have to look at who was stressed, how to define stress physiologically speaking. And when we did that, we looked at women's distribution of first morning urinary cortisol. Cortisol is a marker of the activity of the stress axis. And when you look at the distribution of those first morning urinary cortisol levels, you see a huge amount of variation. You see, for example, this woman with a bimodal distribution of her cortisol levels against this woman with a very clear peak around 2 I think it's picograms per milliliter. You can see that there is quite a bit of variation. And because I was working, all this data come from a population in Guatemala of Mayan women in a very small community, 2,000 people, with very little ethnic, genetic, and social and economic variability. And so I was very puzzled by this amount of variation in the activity of the stress axis, because as we know now, the activity of the stress axis is determinant for a number of health outcomes and behavioral outcomes. And so we wanted to study what the origin of this between women variation in stress axis activity was. As I just said, well, these are two pictures of two women in this study with children that they conceived while we were doing this study in the year 2000. And as I said, very homogeneous population in terms of ethnic background, diet, physical activity, etc. And very little gene flow here too, because as I said, this population can only be reached by boat. So there's not a lot of intermarrying with other communities. So because of that, if genetically they're very homogeneous, socially they are very homogeneous, in their physical activity is very similar, where's the variation in the stress axis coming from? And that's when I started to think it has to come from developmental sources. Something happens in the early life of these women that make them differ so much. I think it's fair to say that at that time in my career, I was very interested in this new movement called Developmental Origins of Health and Disease, DOHAD, that we can also talk about if you want. But this is when working for years on the same system pays off. In the first part of my career, I developed the top of this model where we look at the interactions between the stress and the reproductive axis across many reproductive stages in women. And because of that, as I said, we had collected data from the day kids are conceived for many women. And so I could look at the ontogeny of the stress axis, the development of the stress axis from the absolute very beginning. And until now, we only had information about the effects of that maternal stress has on childhood, puberty, etc. But very, very little. I mean, I would say actually our data set is the only data set, I think even today, that includes the biological specimens from women around the time they conceive their own children. So we realized we were sitting on a gold mine from a researcher's perspective. So we decided to go back to our data set, and we identified 107 moms that I had collected in the year 2000, biological specimens every other day for up to a year, because at the time I was a grad student and had nothing better to do than to live in this beautiful lake in Guatemala and just basically collect every other day urine and saliva from these women. And because I have those urinary specimens, I could look at cortisol levels, I could measure estrogen and progesterone, which allows me to know exactly the day of ovulation of these women. I could also collect human chorionic gonadotrophin, which is the hormone that the pregnancy kits that you buy in the supermarket measure. So I could know exactly the day these women conceived their children. And so we went back to Guatemala, identified 22 mothers and kids that were willing to work with us on a follow-up study and look at how these women's stress axis activity around the time they conceive their children could predict their children's stress axis activity later in life. And because we were able to go in 2013, and considering these kids were conceived between 2000 and 2001, most kids were between 11 and 12 years of age when we were there. And so in order to look at their stress axis activity, we decided to look at these kids' cortisol levels in response to two different challenges. One non-experimental challenge, that is the onset of the academic year, because there are other colleagues that have shown that the first day of class it really triggers a cortisol response in kids, and an experimental response, a 3 stress test, which is a German developed social challenge, which is quite stressful. And look at how kids responded to that challenge. And so the goal of this study was to look at how early post-conception maternal cortisol was related to children's stress axis activity. And we also look at whether or not the relationship that we eventually found could be explained by epigenetic phenomenon. So here I'm going to show you a few results of that study. We're first going to look at the kids' baseline cortisol levels and responsivity to the new academic year. And so here is these kids, in this graph, let me just first say that in this graph, the first day of school is this 0 here, right? And so you can, and these are the first morning urinary cortisol levels for these kids. And as you can see, before the first day of school, the cortisol levels for these kids, the average cortisol levels are a lot lower than right after the first day of school. So the challenge is working. And then you see how they habituate to school and cortisol levels start to go down. So now we can look at whether or not that response is related to their mother's cortisol levels in the first 8 weeks after these kids were conceived. And we here show those results separated by gender, no, sex, gender, because in humans it's very difficult to separate sex and gender. We can have that discussion too if you want. But what you can see is that in average, the response, males or boys' response, was a lot higher than women's response to the first day of school. However, what's super interesting to me is that if you look at maternal cortisol levels after conception, and understanding that the solid blue line here represents the women in the 75th percentile, highest percentile of cortisol post-conception, and the dotted line here in black, the lowest 25th percentile. What you can see is that there is a lot less variation explained by periconceptional maternal cortisol for boys than for girls. In girls, if the mother experience high cord levels right after conception, their daughters also presented a high response to the first day of school compared to the daughters of women who experienced lower cortisol levels when they conceived them. And that was astonishing to us, because we know that males, also adult males and adult females, respond differently to challenges. Then we disaggregated these results by gestational week. So we look at cortisol levels after conception on week 5, after conception, week 2, week 3, week 4. Here, I'm only going to present week 5, just to give you an example of how this works. And here is, so these are kids, separated for week 5 post-conception maternal cortisol levels, boys and girls again. And as you can see, boys do not, the response that boys had to, sorry, this is baseline before school. So baseline before school in cortisol level for boys was not really associated with their mother's cortisol level, but daughters' cortisol levels were positively related. So the higher the cortisol level for the mother, the higher the cortisol level for the daughter. Now we are going to show you, I'm going to show you one of the results of the reaction of these kids to the experimental challenge. So this is a social challenge where it's almost like giving a job interview. So it's known to induce stress. And here is the overall response of all the kids in terms of salivary cortisol to this challenge. So the challenge happens between this T1 and this T2. So this is the baseline cortisol level before the challenge. Here's the challenge, and the T2 is the first response, and T3 and T4 show how they recover from the challenge afterward. And so this is just to show you that the challenge works at activating the stress axis. And again, I'm just going to show you one of our results. And I chose to show you how disaggregated maternal cortisol in gestational week 5, as it happened with the natural challenge of first day of school. For this experimental challenge, we also see an association, a positive association between maternal cortisol on week 5 after gestation and the kid's response to this challenge. In this case, however, we couldn't differentiate between the response of boys and girls. It was only, this result was only significant when we had them all together. But remember, this is a very small sample. So because this is the very first study that we know of this kind, and because the sample is very, very small, these observations have to be interpreted with a lot of caution. However, our results are consistent with our original hypothesis that there may be the variation in stress axis activity that we see in older women may be related to early exposures right after conception. In this case, maternal periconceptional cortisol levels may have an effect in programming the stress axis in terms of its basic activity as well as its response to challenges. And what's to us very, very important is that the timing of the exposure and the sex or gender or both of an individual appears to play a role in modulating those effects. Why do we care about gestational weeks? Because in those early weeks, every day represents a schedule of unique events. And let me backtrack a little. Why is this important to go so early in terms of an individual's development? Because when you start as a 2 cells individual of 4 or 6 or 8, when whatever happens to those early cells will be inherited by daughter cells as those cells divide. And so anything that happens early on will affect a number of biological systems and tissues, having a number of cascade effects in terms of development. In week 5 specifically, we focus our attention on that week because there's a number of cells that will make up the fetal stress axis, the hypothalamus, anterior pituitary, and adrenal cortex, that start to differentiate in that particular week. Now, for those of you who work on the field of maternal stress, you may be asking yourself, well, wait a second, Pablo, what about those protective mechanisms that we know exist that protect the fetus from maternal cortisol, such as placental 11-beta hydroxysteroid dehydrogenase 2, take that, with my accent. Well, we did study that, but I don't have time to go into that right now, but happy I have the slides to answer that question if you want to know in the Q&A section. So I'm going to keep moving forward. Could the link that we found between periconceptional cord and children's stress axis activity be explained by epigenetic mechanisms? The answer is maybe. We looked at the methylation patterns for these children's buccal epithelial cells, so the cells of the interior wall of the cheeks of these kids, and we found that about that over 5% of the changes that we see on 867 sites were related to periconceptional cortisol. And we also found that the kids' HPA activity was associated with changes in 5 CPG sites. Unfortunately, there was no overlap between the 5 CPG sites where we saw epigenetic modifications related to HPA activity of the kids, where there was no overlap with the 867 sites for which we saw methylation changes associated with periconceptional cortisol. So we couldn't complete the loop. We can't say that periconceptional cortisol is affecting the same methylation sites that are affecting HPA activity. But again, this is a very small sample. So, and for these type of studies, you need much, much larger samples. But to us, this is an important pilot study to guide future studies. We also have to consider that there are probably very good alternative explanations, such as shared genes between the mother and the children, as well as the environment that mothers and children share once the kids are born. Okay, then the last study that I'm going to share with you today is a study that we are doing on stress as a modulator of women's aging. A theory known as life history theory in ecology claims that whatever is invested in reproductive effort cannot be invested in somatic maintenance. That means if you have a huge number of kids, you should age faster because all your metabolic energy is being invested in having kids, not in keeping your body healthy. And so the expectation is that reproductive effort should be associated with the pace of senescence. So we looked at that by looking at the number of children women had in that Guatemalan population that I talked about and telomere length. Let me explain just a teeny bit about this. In this Guatemalan population, this Mayan population, at least when I arrived, that's changing, but it was what's known as a natural fertility population. That meant that women did not use contraception, and this is a cultural trait. Kids are seen as God sends, and God decides how many kids you have, and therefore women were not using any type of contraception at the time. And so it's a good community to study how number of children may relate to women's senescence. And telomere length is, I'm not going to go into details right now. I'm happy to chat about it during the Q&A, but this is a marker of cellular aging, although I'm sure that many colleagues will argue against that notion. But at the time, it was controversial, but still accepted. So we look at number of children and telomere length in this population, and we did not find an association. Parity was not associated with the length of their telomeres. Now, we were confused by our results because there's a number of studies that look at women in what's known as weird populations, or Western educated, industrialized, rich, and democratic populations, where they do find an association between parity and telomere length and oxidative stress. So we are not sure why we couldn't find it, but we thought it was important that we reported that. One possible explanation that we could think of was that in this natural fertility population, women who have a lot of children have higher status and tend to receive more social support. And if they can use that social support to free, and social support includes allomaternal care, the care of their children by siblings, by relatives, by friends. And if that help then frees metabolic energy for those women, then those women could be allocating some of that energy to maintaining their body and reducing the pace of cellular aging. And so that's why maybe we do not see within that population that women who have more kids are aging at the same pace as women with fewer kids. And also, it could be that most women in our population have many kids. We did see that women whose kids died age faster, but I'm not presenting those results today because of lack of time. But if you want to know more, please, about either of these things, please ask me on the Q&A. I also have slides for that. Okay, to finalize this talk, I'm going to tell you the three main projects we are working on, and I hope I'm going to remember one more to share with you. So first, we are working on a periconceptional indigenous cohort supported by CIHR. It's known as the Indigenous Healthy, that is based on a 2-I-Seeing approach, and we're working with the Nunchal Tribal Council, which is based in Port Alberni, and that's going fantastic. We're doing really, really good work and learning a lot, and we are hoping that the results of that study are going to help us develop interventions to strengthen kids' development based on resilience that come from ancestral traditions from the Nunchal. We also are doing a similar study in India in collaborations with colleagues from India and Toronto. And we are studying the role of stress, but also sleep patterns and metabolic energy, as well as immune challenges in the pace of reproductive maturation, with a focus on menarche, so the age at which women have their first ovarian cycle, And we are doing that with our colleagues David Sampson at the University of Toronto and Charlie Nunn at Duke University. I have not been timing myself. I hope I am leaving enough time for questions. But before that, I want to thank you for your patience and listening to me rambling for this period of time, as well as to the many, many trainees and collaborators that have helped with the development of these studies over the years. Thank you very much, and I'm very happy to entertain questions if we have time.
Oh yes, we do have time, Pablo. Thank you so much. That was fantastic. Lots of data, and thank you. I was just going to ask people to do the little cartoon reactions because I'm sorry we have muted you all. So yes, if you can share some love with Pablo, I can do the auditory clapping, but you can imagine that there are many animated claps from the audience. Oh, it's really fantastic, great body work. We already have 3 questions from on Slido, and I have a bunch, but I won't take chair's prerogative here. I will ask the questions. So one of the first ones with a smiley, well, winky face is, what about 11 beta HSD? What did you
okay.
find in that?
Let me see if I can. You tell me if I go over time, because I actually have a meeting at 1.
Well, we'll stop at 12:55, so we still.
Yeah. But let me see if I can go to the very end. Okay, here it is. Okay, very, very quickly, there is this enzyme called 11 beta hydroxy steroid dehydrogenase. We call it at home 11 beta HSD2. And what it does is it's produced by the placenta, and it turns bioactive cortisol into inactive cortisone, which is opposite for of what happens with rodents, where cortisone is the bioactive glucocorticoid. So my question was, what is protecting the fetus from maternal cortisol before the placenta develops, right? So what we did is we looked at a number of elective abortion tissue and look at whether or not 11 beta HSD2 was present really early. The earliest specimens that we could find were specimens from week 3. It's very difficult to get tissue from conceptions at earlier ages, right? They're really, really small, and most people don't have, even if they have a spontaneous abortion before week 3, they don't even know. Most of the time it's unknown to the woman, and if you have an elective abortion, it doesn't happen usually before week 3. And so we look at the expression of this enzyme in the cytoplasm of the cells that we obtained, placental cells, and we actually found that this is present as early as week 3. And so as soon as the placenta develops, the protective mechanism exists. But we do understand that the protective system can be overwhelmed. If maternal cortisol levels are really, really high, what happens is that a little bit of cortisol will cross the placenta, and a little bit of cortisol for the mom is a huge amount of cortisol for the baby. What we don't know is what protects the baby before the placenta becomes metabolically active, right? And so anyway, more research to be done there.
I don't know, this is a question I had, so I apologize to the other 2 questioners first, but so you showed some nice epigenetic work. And one question I had, and I think I know what your answer is going to be, but did you split it by sex, gender there? Because you said that there wasn't a good, I've forgotten exactly, but I know part of it, I think you had 5%, greater than 5% change, but it wasn't really linked to maternal cord or something like that. And so I'm wondering if you split it by sex and gender, whether you'd see more of an association in one versus the other, because your other finding was pretty striking, even though males had the higher cohort with school start, you saw a bigger spread, I think, in the girls, and I thought that was really, really, really interesting. So I'm just wondering, did you split by?
No, we did not, because we didn't have statistical power to do that. We need, and this is one of the things that I hope these new periconceptional cohorts will allow us to do, because one of the problems that we have is that there's literally nobody collecting biological specimens from before women get pregnant, and it's understandable, because in order to get 1,000 pregnancies, because the ratio is 25%, you have to start with, for 1,000 pregnancies, you have to start with about 5,000 women who are, and follow them for about a year. So it's not cheap, and you have to collect, and you know, these are women who know me for many years and are collaborate. But if I go and I say, Lisa, will you pee for me every day on a cup for a year? I don't want to know the answer, because it's probably not going to be a very nice.
For you, maybe, but you know, for other people, no. And I'm too old now, anyway. So the other question, though, I can appreciate that. I still think, maybe I shouldn't say this out loud, but I still think in in your dark room, sometimes it's good to just split it by sex, even sex / gender, even if you have a low end, just to see. But that's
yeah, I think we did it, and I can't remember what we found.
What you found.
You know, this is Cindy's work in our labs, and Cindy, for everybody, Cindy is a former PhD student from Lisa, and my former postdoctoral fellow. We can ask her. I can't remember at this time.
Because she probably did do it in the darkness of her.
I'm sure she would do that. Yeah.
All right. So we have a couple more minutes here, but do you think there was an element? They're both based on the stressor, which I really like the naturalistic stressor versus the TSST. I really like that approach. So do you think there's an element to attending school that might have been more stressful for the girls versus the boys? And I'm wondering, I mean, one way you could probably address this is, did you see a difference in cortisol response in girls versus boys attending school?
Well, I mean, boys overall had a higher court response, but the spread was more in girls, which makes me believe, but this is all speculation at this point, makes me believe that there may be other traits in girls that are determining how stressful it is for them to go to school. And you know, that goes to cultural and gender studies that I wouldn't dare to speculate on that, because I'm you know, at the end of the day, I am a biologist. I'm an anthropologist, but I'm a biological anthropologist. So I don't know I don't know enough to venture into speculations on cultural or gender aspect. But I'm happy for anybody that are working on those fields to come and talk, and I'm happy, open to collaborations. Oh, by the way, let me put a plug for, we have a project on stress and perimenopause that is going really slowly. So if there's anybody interested in those data, it's going slowly because the funding is basically, we are stealing from other projects to get that going.
Oh, I know that feeling.
but the data is there, and we can write more grants or you know, if somebody wants, you know.
I know a few people that would be really interested in that, so maybe we can talk offline. And obviously, if anybody's on the call, please reach out to us, because that would be fantastic. Oh, Cindy Barha, because she can't say anything, but she just texted me and she said it didn't change the results. So there we got the answer.
I'm glad. Hi, Cindy.
Great. Hi, Cindy. Thank you Thank you so much for the answer. I'm just wondering, in follow-up to that last question, what about puberty? Like, so were the girls, and I mean, obviously girls go through puberty earlier than boys, but it was age 11, so that is right around the time. I'm wondering if the girls had gone through puberty and the boys hadn't, or did you take measures of that or at all?
Yeah, we are working on that right now. So a PhD student, Amanda Rowlands, I don't know if Amanda is online now, but she and another student, oh, Amanda got the award from the Women's Health Research.
Oh, great.
2 years ago. And then Ana Paula Costa, a grad student from Brazil, both of them are working on that topic. We have some results, but I'm not going to steal their thunder. It's their project. But we are finding really interesting stuff in terms of the timing of menarche, as well as the pace in terms of the maturation of their reproductive axis. Because once you have your first cycle, then you go through a period where cycles are very regular, where you might have very frequent anovulatory cycles, and so we're studying the relationship of those of that timing and that irregularity with challenges up to that point, right? From periconceptional maternal cortisol levels to anything that happens thereafter. Very interesting.
Oh, that's great. Well, we'll have to have you back. So the last question we have, and then we'll let you go a little bit early, is regarding, this is the problem with having these Zoom meetings, right? Because you book things back to back, but then you realize you still need some bathroom breaks.
Yeah.
Regarding female child mother dyad port levels, do you think the type of stressor is important? So social stressor, such as school starts versus the psychological stressors such as TSST.
Sorry, I didn't get the question.
So I think you sort of answered it within your presentation, and that is, did it matter whether it was a natural stressor with start of school versus TSST?
Yeah, no, it didn't. I mean, that was what was surprising is that, and being very encouraging is that whether it was first day of school or it was this experimental challenge, which is it's actually very punctual. So it very different challenges, right? One is, oh my God, the whole year starts and this is going to last 9 months of my life. I'm going to go to school and I have to deal with, obligations and boys around me. And the other one is, oh my God, right now I have this exam in front of me with this unknown these unknown people. They're going to ask me questions. I don't know what they want from me. They are white people. I don't know them. One is it's kind of I'm in front of a chronic challenge. And the other one is I'm in front of a super high, intense, acute stressor right now. And yet the link with maternal periconceptional cortisol was pretty much the same. And the effects, I didn't show you all the results, but the differences between boys and girls was pretty consistent, even at the periconceptional week level. So that was, I think that there is a lot to do research on right there.
And then when you were talking about the telomere link, the other thing that I was thinking about is and this is just from Wendy Robinson and working a little bit with her as well, but the epigenetic clock, like, have you thought about doing some experiments looking at that instead of telomere length?
Yeah, correct. We actually did this work with telomere length is with Wendy.
Oh yeah,
yeah, and as soon as we published it, and she told me that was going to happen, but at that time, I had chick swabs, that's all I had, and people were working on the development of these clocks. I would love to do something with clocks. There is Wendy, there is Mike Cover, there is Megan Jones, who are doing Caleb Ryan. There's a number of really cool colleagues doing work on that topic. It's not my topic, but I welcome anybody that wants to use the samples that we have for that purpose, because it's within the topics that women have agreed in this population that we could study. So, and I think they will be very interested in finding out.
Yeah, it'd be really great. Okay, we have one last question really quickly. How do you combat loss to follow-up? So you have this fantastic Mayan population. So do you have a lot of loss in your follow-up work, or and how do you deal with that?
No, that's the fantastic thing of a remote population is very low attrition, because the people don't, people tend to not move much. And, I spent almost 2 years. I spent a huge amount of time before I started the study, and almost over a year collecting samples. So I developed, with some of those people, we're family. Actually, the meeting now at 1 is with people from Guatemala, and so and that and if I have to be fair, probably they see me as a resource, but not just an economic resource, because I'm their advocate when they need something from the government, etc. So it's a very different situation than, I joke, if I go and ask you, Lisa, if you will pee for me for a year, and that's the relationship completely different, because I don't know how to explain it. It's a very small community. It's less than 2,000 people. So the attrition was not an issue.
That's okay.
Now, in 2017, we did the last field trip, and there are a couple of women who have been postmenopausal for almost a decade. And when I go and I ask them for urine, they tell me, Pablo, don't you think it's enough? I mean, that, you've been doing this for a while. So I had like 2 women say, no, I don't want to do this anymore. And that, totally fine. It's just like, it's their choice.
That's great. Well, you've obviously invested a lot of time, and so that also pays off. So I just want to make sure you make your next meeting. So thank you. I just want to say thank you again. If people can join me in their clap reactions. Thank you so much to you. It was a fantastic talk and really fantastic work, and we look forward to hearing more. And if people are interested in collaborating with Pablo, please reach out to us and we'll connect you, or to Pablo.
Yeah.
So thank you so much, Pablo. I hope you make your next meeting.
Thank you very, very much. It was a pleasure. A lot of fun. Thank you, Lisa, and everybody else.
Bye. Bye, everyone.